Hi, I'm Alison Shapcott. I'm representing a team of researchers, and we've been using molecular ecology approaches to enhance our understanding of ecosystem renewal. Molecular markers are leading to new opportunities to address new ecological questions. Population genetics uses molecular markers to look at mating, dispersal, and relationships and diversity within individual species. DNA barcoding enables us to have standardized genetic measures that enable a kind of community genetics to investigate relationships and diversity of different communities. DNA sequence reference libraries enable the identification of unknown samples. And metabarcoding and eDNA enable the identification of unknown samples that are mixed, for example, in animal guts or in the environment. The ability to use molecular tools for ecological questions depends on the building of verified reference databases of standard sequences of DNA, that is, of a particular section of DNA. DNA barcode reference sequence libraries have been building for the last decade, and they're fast growing, but there's so many different plants that we still have lots of gaps, and particularly for Australian taxa. There's new sets of universal genetic markers being developed for plants, but these are still in their infancy, and so the reference libraries are quite small. We've been building DNA barcode reference libraries for rainforest plants and heath plants in southeast Queensland to use to investigate ecological questions. So this talk will profile some of the examples from our work where molecular approaches have been used to investigate ecosystem renewal. So the first example I'm going to give is of the recent bushfires that burnt the rainforest in southeast Queensland. We had already investigated the phylogenetic diversity and relationships of the different rainforest types and the different ecosystem types and subregions of southeast Queensland. We overlaid this with the fire extent mapping. We were able to determine that the subregion that had the greatest amount of rainforest burnt compared to any other region, as shown on this map with the yellow, was region 12.1. Now, unfortunately, this region, which had approximately 20% of its rainforest impacted by fire, is the subregion that has the second highest phylogenetic diversity of all of the different subregions containing rainforests in southeast Queensland. We also found results completely contrasted with what we expected. We thought that the wetter rainforest associated with refugia that are typically phylogenetically evenly dispersed across the possible species present, we thought they would burn less than the phylogenetic clustered or drier, more recently expanded rainforest types. But we actually found that significantly more rainforest of the moister type burnt compared to the wetter type burnt. So this has big biodiversity implications. We also invested central Queensland rainforest communities. These are very poorly studied. And we did quite an extensive survey of more than 500 sites. And our results indicate that the central Queensland rainforest communities are not really grouped into very many distinctive groupings, and they're more consistent with a kind of corridor between the tropical and subtropical rainforests. But we did find some groupings. You can see in this diagram, in the area encircled, that sites that were in coastal sand with low rainfall were phylogenetically more similar to each other. They're clustered, as indicated by the circles. We found that only 6% of our sites were significantly phylogenetically even, so it doesn't look like there are big areas of potentially refugial rainforest. We found 17% of the sites were, on the other hand, phylogenetically clustered, which is suggestive of some sort of environmental filtering. We also used DNA barcoding in Sunshine Coast heathlands to enhance our conservation priorities. You can see on the phylogeny shown here, the green represents the heath species. The rest of the phylogeny is the combined rainforest and heath. And you can see that the heath is very unevenly distributed. It's clustered in that phylogeny. We also found that the overall Sunshine Coast heath has lower phylogenetic diversity. And we found that 8 of the 9 regional ecosystems that are representing heath were significantly lower in their phylogenetic diversity than you'd expect by chance. However, only 6 of the 80 sites that we studied were phylogenetically clustered, even though overall the taxa is clustered. And these are represented by the red dots in the diagram. But what's perhaps more interesting are the clustering of the green dots that represent significantly even sites in our study. Now, these are often associated with refugia and could be heath refugia. In this case, they're phylogenetically clustered, so they're similar in composition, but they were geographically dispersed across the Sunshine Coast. On the other hand, their environment was very similar. They were found in the wet heaths. So this suggests more of a refugial environment than a refugial location. We used metabarcoding to investigate the resource competition between the pollen carried by honeybees and native bees in subtropical heathland. And what we've got represented here is a network diagram with the bees on the top and the plant species on the bottom. And we can see that there is quite a wide overlap in the diets of the pollen by the different bee species. So lots of species of plants are used by different types of bees. And you can see the niche overlap index is quite high. We found that both stingless bees and honeybees have quite wide foraging patterns across multiple species. But we also found that individual honeybees can carry a greater number of different pollen species on the single bee. But on the other hand, native bees forage across a wider range total species. We also used metabarcoding to investigate the floral resources used by native bees. And we compared 2 ecosystem types, a macadamia orchard and a nearby native forest habitat. And we were able to identify at least 118 different plant taxa that are utilised for different types of resources by the native bees. Now, the different colours represent different parts of the beehives that we sampled and tested using metabarcoding. So we can see the different uses of the different plants. And some plants are used by the same bees for different purposes, and some different types of bees use different types of plants. And some plants were found across multiple different uses. We found that 20% of the plant species were used more than 70% of all the resources used. And the common ones between these 2 ecosystems were mostly trees and vines. We were interested in the vines. And some of the commonly used trees were Eucalyptus racemosa and 2 different species of Lophostomen. We also used molecular approaches to investigate the mechanism of action of Phylinus noxious. Now, Phylinus noxious is a plant pathogen. It's a fungus that causes brown root rot disease, and it affects a lot of street trees as well as native forests. Now, another fungus, Trichoderma species, are widely used as biological control agents to control the growth of this pathogenic fungus. And one of the key mechanisms of action of Trichoderma against Phylinus is that we get an increased expression of the fungal cell wall degrading enzymes, and these two enzymes are particularly noted. And what we found, as you can see when we grew them together, that we had an increase in the expression, particularly of the chitinase. It was 8 times more expressed when we had the presence of these together. So molecular tools are now quite widespread in ecological research, and they can greatly enhance otherwise quite difficult areas of study, and they enable a diversity of new questions to be asked. However, there are an increasing array of different kinds of tools available, and while this allows a diversity of different types of questions and applications, it also means that ecologists need to take the molecular advice regarding the appropriate tools for the questions of interest, because it's not a one-size-fits-all. And the nature of the ecological questions that will be able to be addressed by any particular study will be constrained by the reference libraries that are available. So ecologists could really consider in the early stages of their projects funding the development of project-specific sequence libraries, because this will mean that you can get much better resolution and much better results for your study. We would like to acknowledge a great many people and organisations who funded these different projects, and particularly this reference library databases. And we also want to acknowledge the custodians of the many lands that this work was conducted, and pay respect to the elders past and emerging.